Let's talk about Mendel's laws of inheritance. So these are two laws: law of segregation and law of independent assortment. Previously, geneticists used to think that the characteristics which are expressed in organisms, when there is a cross between two parents, and if they have different characteristics, then the characteristics get blended, mixed, and gives a mixed or blended characteristics in the next generation. uh, this is what Mendel was able to disapprove with, and gave the law of segregation. And the second one is law of independent assortment of the factors of inheritance, which I'm going to come to later. I will be talking about both of this later. And now, first is monohybrid cross. It is a cross between two parents of different a cross between two parents, which is done to analyze a particular one characteristic, not trait, one characteristic. Now, the law of segregation. According to it, the hereditary factors are discrete and do not blend when present together. They do not get mixed. So let us first understand a few terminologies. The parent generation, then the next generation is known as filial one generation, or this first filial generation, and then F2 generation, or the second filial generation. So what Mendel did is he took a pure breed of tall and a pure breed of dwarf plant and crossed them to produce a tall pea plant. And when he crossed the F1 generation plants, tall plants, he found all tall plants, and when he crossed between tall plants of the F1 generation, he found 25% of dwarf and 20 and 75% of tall. So what did that mean? According to him, it meant that that these gametes, or these hereditary factors, do not get mixed, do not blend, but remain discrete. That is, they undergo segregation but not blending. That's why in the next generation they are able to produce 25% of dwarf and 75% of tall. And his explanation, or the model he proposed, was this: that let us if we take the example of tall, then one minute. If we take the example of tall, then uh, then it is possible that this contains tt and this contains small t, this dwarf, and in the gamete only one t comes, and in the other one only the small t, and it results in t small t. But out here there is a dominant trait, that is the tall one, so it gives, but it results in all tall. But when there is cross in between f1 generations, so, then this kind of combination occurs, so resulting in 25% of dwarf and 71 and 75% of tall. So this is it, and this is the law of segregation. And now let us understand a few things, that is a few terminologies. Test cross: it is a cross between the filial one generation offspring and a recessive pure gene. So this is S cross, where there is a cross between the F1 generation offspring and the recessive pure. This helps in understanding if this so this to determine if this is dominant, or it is a pure tt small large tall t, or a dwarf genotype. This can be done, because if it is tall, then it would only result in tall offsprings. But if it is tt, it is heterozygous, then it would also result in dwarf ones. 50% would be dwarf and 50% would be tall. Now next, the back cross. So this is any kind of cross where if the genotype of any, this is a this is back cross, where there is one F1 individual and offspring and a cross with its parental genotype, and it results in uh, sorry, this is not. And this kind of crossover is known as back cross. This is what test cross. So as I have already said, so now the thing is that this test cross is actually a subset of back cross. Since out here it can be any parental offspring. In case of back cross means what? Back cross with a parent. And test cross is for a test to determine if this F1 generation one is a hetero contains um, um, it is heterozygous or homozygous. Determine that one. So it is crossed with a recessive pure one, and if it results in all pure, then it is homozygous stall. And if it yields some offsprings, that is 50% tall, 50% dwarf, then it should be heterozygous. So this is it for now. And this type of cross is known as Punnett square. To find out all the combinations which are possible, it was proposed by R. Punnett. And now let's dive into the laws of independent assortment given by Mendel. So for this we need to do a dihybrid cross, which means a cross focusing on two different characteristics. And what does independent assortment mean? It means that a gene which occurs independent of the a gene gets assorted in the gametes during gametogenesis, independent of other non-allelic gene sets, and thus it results in new combinations of characteristics in the offspring. So how I did that? He used pure yellow ground G seeds where there was YY, capital Y, and capital resulted in this gamete, and green wrinkle seed, small y, smaller, resulted in this gamete. Thus, it resulted in yellow and red seeds, but these were actually heterozygous in both cases. Thus, when their offsprings are produced, they produced genotypes of the ratio this and phenotypes of the ratio this, and this is in accordance if another Punnett square was made. And so these ratios are in accordance if there was independent assortment, that is R and R had the tendency of getting assorted into gametes independent of each other. So from this he gave this, that yes, there is independent assortment. And in case of dihybrid test cross, where the offspring or the F1 generation offspring is crossed with a parental P generation recessive one, then it results in 1:1:1:1. So from this, if it gets 1:1:1, that means that the if that the dominant one of unknown genotype is having heterozygous nature of things. Heterozygous genotype. And so this is it. And test crosses can also be actually used to determine whether these two genes are actually linked or not, and as well as if the underlying genotype, if an allele penetrance is more less than 100%. So what is penetrance, we'll know that later.